Dear students, in this presentation I am going to give you the simplified and overall understanding of the complex process of eukaryotic mRNA splicing. Myself, Dr. Wasim, Assistant Professor of Department of Biochemistry, Ramaiah College. So eukaryotic genome is basically complex compared to prokaryotic genome, and the complexity is mainly attributed primarily because of the nucleosome structure. So in eukaryotes, the genome is having DNA along with the nucleoproteins called histones. So the DNA is winding around the histone protein in a way that the important accessible regions like promoters, enhancers, and operator elements are buried within the histone core. So any process requires the access of these promoter elements or enhancers and operator elements has to be started with the remodeling of these systems. So this is very important regulation in case of eukaryotes. The accessibility of the important regulatory elements, the promoter, for instance, for the transcription to start, the accessibility of promoter elements is itself an important regulation. And another point of complexity in case of eukaryotic genome is because of the presence of split genes. In eukaryotic DNA, the genes are not continuously coded just as in prokaryotes. Rather, it has got coding exons which are separated by the relatively longer stretch of intervening sequences which are non-coding introns. So during the process of transcription, the initial hRNA form is having both exons and introns, and they undergo the process of splicing, which results in the spliced exon, which we call it as mature mRNA, which is ready for undergoing the translation process, and leaving group is called intron lariat. So this splicing, which is taking place as a complicated event in case of eukaryotes, is taking place preferentially at particular sequences. Those signature sequences will form the splicing signals. Splicing signals in hnRNA are very specific, that are present at exon intron borders and intron exon borders, and there is a branch point site. These all together acts as splicing signals. So the 5' splice site, which is also called as donor site or exon intron border, this has got the borders by the guanine for exon and another guanine for the intron. And there is an adenine which acts as a branch site. And at intron exon border there is again another guanine for intron ending, and for exon starting there is another guanine. This is called 3' splice site or acceptor site. The chemistry of splicing basically involves the two trans esterification reactions. The first trans esterification is initiated by the attack of the branch site adenine hydroxyl group onto the phosphate group of the guanine that is present at the donor site, the intron guanine. And this first trans esterification reaction results in the formation of intron lariat, whereas second trans esterification reaction results due to the attack of the hydroxyl group of guanine present at the exon, the freed exon, onto the phosphate group of the next exon of the guanine present at the 3' splice site. So at the end of the two trans esterification reactions results in the formation of intron lariat and joining of the two of the exons. So this whole together comprises the complex process of splicing. So splicing mechanism is basically nothing but the SM2 type of nucleophilic substitution reaction that is mediated through a very unstable pentavalent state of phosphate of adenine. So this is unstable and transient form of this intermediate product in the lariat structure. And this splicing mechanism does not require energy. However, splicing process is ATP dependent, which is used for the assembly of spliceosome complex. Spliceosome complex formation proceeds sequentially in a stepwise manner, which is a very critical event that eventually results in heterogeneous nuclear RNA to form mature mRNA. So this is basically mediated by the participation of RNA and protein that exists as a complex known as SNRs. So altogether spliceosome machinery includes about 150 proteins and 5 RNAs that together forms a spliceosome complex of about the size of a ribosome. So SNRs include 5 RNAs that are U1, U2, U4, U5 and U6. These are collectively called as small nuclear RNAs, snRNAs. So these RNAs will be in complex with the proteins and are known as small nuclear ribonuclear proteins, snRNPs. These are also pronounced as SNRs. So RNAs basically can align with the specific splice site signals on hnRNA. So these are basically the RNA-RNA interactions responsible for the specific recognition of particular splice site by the particular snRNAs. So that is the reason how snRNPs, the different type of snRNPs are binding to specific splice site signals. So now the steps of spliceosome complex formation and splicing is a sequence of events that starts from the binding of U1 snRNP along with branch point binding protein and U2 AF6535. So this binding to hnRNA results in the early complex formation. This is followed by the U2 snRNP that actually replaces the branch point binding protein which is bound to the branch site adenine. So this results in leaving of branch point binding protein and U2 will replace the branch point binding protein at the adenine site. And the third step includes the addition of U4, U5, U6 as tri-snRNP that binds to the complex, and the U2F6535 will be the leaving group at this particular step. This is followed by the addition by the removal of U1 and U4 snRNP, and U6 and U2 interaction in the group are going to form the critical RNA-RNA interactions that are basically responsible for holding the exon and intron at the reaction center, and they are going to be holding the two groups which are already freed. So this finally results in the spliced exon and the leaving intron lariat. This illustration depicts the sequence of steps involved in spliceosome complex formation that results in splicing. The initial event includes the identification and binding of U1 snRNP at 5' splice site and branch point binding protein along with U2AF6535 at branch site and polypyrimidine tract. This results in the formation of early or E complex. This complex then undergoes the replacement of branch point binding protein by U2 snRNP, resulting in the formation of complex A. Complex A then undergoes the addition of U4, U5, U6 tri-snRNP particle with the removal of U2AF6535 from the spliceosome assembly. This results in the formation of complex B. Complex B formed will then undergoes the rearrangement in such a way that the U1 and U4 SNRNPs will be removed from the assembly, and the alignment of the SNRNPs will be in such a way that U6 and U2 SNRNPs will align in its proximity. So U6, U2 RNA-RNA interactions are very critical for the proper splicing to take place. U6 is responsible for holding the cleaved exon, and U2 will bind and stabilize the intron lariat. Thus, this C complex, the terminal complex, finally leads to the formation of the spliced exon or mature mRNA, which then undergoes translation process to give rise to the final protein product, whereas intron lariat subsequently undergoes degradation. This completes the overall understanding with the salient steps involved in splicing that is responsible for formation of mature mRNA from hnRNA in eukaryotes. I have taken Molecular Biology of Gene 7th edition by Watson to prepare this material. I thank all the listeners for listening to my video. Thank you.